William Shakespeare. William Shakespeare, in his play The Tempest, had one of his characters, a character called Caliban, curse the master who had him enslaved him, saying, all the infections that the sun sucks up from bogs, fens, flats on prosper fall, and make him an inchmeal with disease. The curse that Caliban was talking about was a deadly fever. It was recognized in many parts of Shakespeare's England and other places in the 1600s, but one that was more prevalent in the tropics. The fever, of course, is malaria, a disease that has plagued humankind ever since we probably knew about disease. And the great physician Hippocrates, who lived in ancient Greece, recognized these deadly fevers. He described them, the intermittent chills and fever that people had. But more than 2,000 years ago, Hippocrates could not understand the nature of this disease. And you may recall from a previous lecture that we had here in this course. Let's see if we can turn on the slides. You may recall that this fever was called marsh fever because it emanated from marshes or bogs. And here is the fever agent emanating from the water. And what's happening is that the shepherds, farmers are falling down with this bad air. Because you remember that malaria, the word itself, means bad air. And so the belief was that this disease was associated with bogs and swamps, and it was caused by the bad air. Now, as you probably already know, the earliest recorded history of malaria did not yield to an understanding of the disease until 1880. And at that time, a French physician named Alphonse Laveran discovered in the blood of a soldier, and this was in Algeria, he examined the blood of this patient who had these fevers, these malarious fevers, and he examined using a rather crude microscope. He examined a drop of blood under the microscope. And what Laveran saw was shown in the next slide. And what Laveran saw, can we focus it just a wee bit? Oh, it's getting better, get better. Oh, pretty good. How was it for you? Okay. So what Laveran saw were these globules inside the red blood cells, and eventually these burst out of the red blood cell, and he saw these whip-like filaments coming out, and he also saw these crescents. And he recognized quite clearly that this was an animal parasite in the blood of this patient, and he described the etiologic agent for malaria. No, I'm not Monsieur Laveran. No, I am quite a different nationality. I am Surgeon Major Ronald Ross, and I'm the Knight Commander of Bath and Knight Commander of the Order of St. Michael and St. George. And presently, I'm the Professor of Parasitology at Liverpool University, School of Tropical Medicine, and I am the recipient of the 1902 Nobel Prize for Medicine. And I received this Nobel Prize for my work on malaria, about which I will speak shortly. Now, malaria, as most of us know it today, thanks to Monsieur Laveran, is a disease of the blood. The chills and fever are due to the bursting of these red blood cells by the parasites. And this triggers a body reaction, which then elevates the temperature, and the person goes into this fever, followed by a chill. And in some cases, of course, there's severe anemia associated with the destruction of these red blood cells. Sometimes death occurs. But even when death does not occur, this disease is debilitating. And individuals cannot work, and they must remain in bed. And so consequently, malaria saps the strength of the individual who is infected with this disease. Now, you may be surprised to know that even today, 2,000 years after Hippocrates, there are more malaria cases on the face of the earth than ever before, maybe 300 million. And there are about 2 to 3 million deaths every year, mostly in Africa and mostly in children. So you see, identifying, as Monsieur Laveran did, the causative agent of this disease did not ipso facto eliminate this disease. Indeed, you will be as shocked as I am to know that there is no vaccine, and even some of the drugs are not as effective as they once were. But I will explain this at the end of the lecture, why we have more malaria today than ever before. I am British, but I was born in India on May 13th, 1857. And I look pretty good for somebody that was born in 1857, you will admit. I'm the eldest of the Ross children. My father was an officer in the British Army that was stationed in India. And remember, at that time, in 1857, India was a part of the British Empire. It was there that my family and I first experienced malaria. My father had it. We had to take quinine. You probably call it quinine, but we called it quinine. And we took it with regularity to ward off the infection. Sometimes it worked, sometimes it didn't. Now, as a boy of 8, as was typical of the colonials, as a boy of 8, I was sent to England to school to be educated. I was a dreamer, and although I liked mathematics, and I also liked writing, I wandered around the hillsides, around the school, collecting butterflies, collecting insects of all sorts, collecting animals. I was observing plants, and so on. And at Spring Hill Boarding School, which I attended subsequently from the age of 12, I began to write some poetry. I painted watercolors and thought of becoming an artist. But my father, who was this military man, he had other ideas for me. And he said, "You study medicine in London." And of course, in those days, one listened to one's parents. And so I began my medical studies in preparation for joining the Indian Medical Service. I was not really a very good student, and not because I was lazy, but because I had so many other interests. I couldn't concentrate on medicine. I did pass my examinations. I have to admit, I did have to take it twice in order to pass the exam, and I was, in fact, employed by the Indian Medical Association. I first worked as a ship's doctor and then was posted to India. And in India, I was in Bombay and Madras and Bangalore, and at most of these posts, life was exceedingly dull. Oh, we saw some patients in the morning, and sometimes there was an evening where one had to perform service, but most of the days and afternoons were free, and I was free to play my violin, to paint my watercolors, to study languages. It was really quite nice. And I wrote novels and poetry. Most of it was totally unappreciated, and I could never get it published at a publisher's expense, so I had them published at my expense. Now, on my first leave home from India, leave to England, of course, I met and married my wife, Rosa. And then I returned to England on a second leave in 1894. By that time, I had spent 13 years in the Indian Medical Service. My scientific accomplishments were nil. I wrote a few papers on malaria, and I had them published in the Indian Medical Gazette, but I failed to confirm Laveran's findings. I couldn't even see these parasites in the blood with my microscope. And I even claimed that malaria was an intestinal infection. What a fool I was. Everything I had turned to was for naught. Now, one of the most perplexing problems with regard to malaria at this time was, how does one become infected with this parasite? And then in 1894, I remember the exact day, it was April 9th, I visited with Dr. Patrick Manson. Manson, who's sometimes called the Mosquito Man, he had worked in China, and he had studied elephantiasis, which is caused by a worm. Filariasis is the technical term. And Manson had shown that mosquitoes were the transmitters of this disease. And Manson had been, after this posting in China, had returned to England, where he became a professor of tropical medicine in London. He was an outstanding microscopist. And he showed me that Laveran's parasites were peppered with this brownish-black pigment. And he showed me exflagellation, these things that shot out from the red blood cell, you remember, from these crescents. And he became my mentor. And one day, as we were walking down Oxford Street, he said to me, I have formed this theory that the mosquitoes carry malaria. He had a very high-pitched voice, this Manson man. And he said, they get into the mosquito's stomach, and they shoot out those whips, you know. And the whips get into the mosquito's body, where they turn into a spore, and like those of anthrax. And then the mosquito dies, and it falls into the water. And when people drink this soup of dead mosquitoes with these spores, they get malaria. This was Manson's theory. Now, it might sound a bit far-fetched, but it's really not quite. As I mentioned earlier, the malaria had been associated with swampy bogs, fens, an area where there was this sort of wet sogginess, bad air that emanated from these low-lying areas. And this was known, as I've said, for thousands of years. And, of course, the name of the disease takes its name from the air above these bogs and fens and swamps. So it was believed that the vapors that came from the swamp produced the disease. And, of course, before Laveran's discovery, that's the way most people believed one acquired this disease. In fact, by the time the germ theory had been elaborated, namely that germs cause disease, Robert Koch had two of his associates studying malaria, and these associates sampled the water in these swampy areas, and they found a bacillus. And they found this bacillus also in the urine of a patient that had malaria. And they at once named this bacillus Bacillus malaria. And they claimed that it was the cause of this disease. But no one could ever reproduce this work. And so it was soon forgotten. Now, I left England in 1895. March of 1895, I reached Bombay a month later, so you can see that the trip from England to India took about 4 weeks, and I had a mission. And this was the mission that Manson had put me on. I wanted to find the means of malaria transmission. How was this disease transmitted? And in June of 1895, encouraged again by Manson, I set up an experiment. I remember Manson said that these mosquitoes roll up and die. They form a spore, and then a person drinks this mosquito-laden water, and they become infected. So I set up an experiment. We had some water. We had some dead mosquitoes that had fed on a malaria patient. These mosquitoes died, and I fed this water to a volunteer. Now, this man actually became ill. He ran a small fever, but when I examined his blood, there were no crescents in it. And so I began to think that maybe Manson was not quite correct. Remember, Manson's theory would be, well, you have to have ingestion. Ingestion means eat. I began to think maybe it was inoculation. That is, it had something to do with bite. Now, at this point, I must tell you a little bit about what we knew concerning malaria and the development of this parasite in the blood when I began this experiment. So let me show you what these parasites look like. You've seen some of this before. Maybe I have. Next slide, please. So here is the development of the malaria parasite. Here is a red blood cell. Parasite is inside the cell. It gets bigger, 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 bigger. And eventually it gets so big that its nucleus begins to divide. And it begins to produce this pigment, this telltale pigment called malaria pigment. And Manson had encouraged me. He said, follow the trail of the pigment. And that's what I was going to do. And then, of course, the parasites eventually destroy the red blood cell, and they cause the anemia by destroying these red blood cells. But also present in the blood are these banana-shaped. The cells, these are parasites. They give the name falciparum to this parasite, and these are the sex cells. And remember that some of them shoot out the whips, and these whips are the equivalent of the male gamete in other organisms, namely the equivalent of sperm. And they also have this pigment in their bodies. And remember that when the red cell bursts, that's when the fever results. This is essentially what we knew about malaria when I began this work in 1895. Now, I immediately began to work with patients whose blood contained these crescents. And the first thing that I had to do was get mosquitoes. So I collected mosquitoes, I had them lay eggs, and eventually they had larvae, these little worm-like forms, and eventually they matured into adult forms. So I allowed these mosquitoes that I had bred in my laboratory to bite individuals who had crescents in the blood. Now, at this time, there were about, the estimate was 2,500 different kinds of mosquitoes, and there were no good ways of describing the differences between mosquitoes. Most of you probably can't describe the difference between the mosquito that bites you. Usually you go, okay, that's a dead mosquito, or that's a live mosquito. But you have to be able to distinguish the different kinds of species. Now, when I found that almost no matter the kind of mosquito, if it fed on these crescents, you would get these whip-like forms in the stomach, but no further development took place. And so I failed time and time again to get any further development in these mosquitoes. Now today, I understand, as I did not then, that I was using the wrong kind of mosquitoes. There were three kinds of mosquitoes that were particularly interesting. One was the gray kind. The other was called the brindled kind. Brindled is a fancy word for striped. And then there was a dapple-winged mosquito. And dapple, as you know, means that it has spots. No, I worked with the gray one, which is Culex. And Culex is not a transmitter of malaria. The brindled one, we know today, the stripy one, is called Aedes. And Aedes is a transmitter of yellow fever. No, the dapple-winged one was the one I should have focused attention on. And this one we know today as Anopheles. Hindsight being 2020, of course, it's easy to see why this is true. I wrote to Manson about my lack of success. And of course, each mosquito dissection, looking for these parasite stages in the body of mosquito, you have to remember what the size of a mosquito is. I want you to think about making a dissection of a mosquito. You have to do it under a microscope. And of course, you don't have any air conditioning or fan or anybody fanning you. Why? Because if you go like that, the mosquito is gone. So you have to work in still air. And of course, the weather in India was really quite warm and very humid. Now, Manson, every time I wrote him, he responded with words of encouragement. And then in August of 1897, I was now 40 years old and had spent 17 years in the Indian Medical Service. I turned from the incompetent mosquitoes, namely the gray and the brindled, I turned to the dapple-winged one. And on August 16th, 1897, my assistant brought me a bottle in which the mosquitoes had been growing and laying eggs, and then they developed eventually into adults. And he brought me about a dozen big brown fellows. Well, they really weren't fellows. They were really ladies. Because as you probably know, only the female mosquito takes blood. And so it was the ladies that we were really interested in feeding on these patients. Now these mosquitoes, and I'll show you what one looks like. Can we have the next one? Thanks. This is an Anopheles mosquito. You notice that it bites at an angle. Here is the surface of the skin. And you can also see in the body of this mosquito this nice red material. So the body has been filled with blood. And this is the Anopheles mosquito, the dapple-winged mosquito that is the transmitter of malaria, and which was the basis for my discovery and Nobel Prize. Now, what I wrote in my memoirs is that about a dozen of these big brown fellows with fine tapered bodies and spotted wings were hungrily trying to escape from the gauze that covered this little flask. But the angel of fate had placed his hand on my shoulder, and amongst the 50 different kinds of Anopheles mosquitoes, we had picked the right one. These Anopheles, by the way, bite at night. And they also land on the wall at an angle. Now, I've caught one of these in this net here, and this is somewhat larger than life size. So here is an Anopheles mosquito, more or less. If I break this, Dr. Platzer will kill me. Okay. So there is the mosquito, and they land at this angle, and of course use this proboscis and suck the blood. This one is at a different angle because it wouldn't stand. So it was August. My mind was blank with the August heat. The screws of the microscope were already rusted from the sweat from my brow, and my hands were also sweaty. Just think about how miserable it must have been. The eyepiece on the microscope was cracked. And then I fed these Anopheles on a volunteer who had crescents in his blood. His name was Hassan Khan. And only 3 of these Anopheles mosquitoes were left on the morning of August 20th. One of them had died and swelled up with decay, that is, the mosquito. And at 7 a.m., I went to the hospital to look at the patients. I attended to some correspondence, and then I dissected the dead mosquito. And of course, there was nothing of any interest in it. Okay. About 1 p.m., I determined to sacrifice the last Anopheles mosquito. I asked myself, was it worth bothering with this mosquito? After all of these failures. But I took the position, any job worth doing is worth doing well, and the dissection was excellent. I have to admit, I went carefully through all of the tissues that were now so familiar to me. I had dissected probably 1,000 mosquitoes during this time, or maybe more. In fact, I still had the passion when I did this dissection as if I was going through a palace, looking for some jewel, a room that contained the jewels in the palace. But then I thought, well, maybe this is going to be an empty palace. It's going to be a failure. And then the tissues that I had yet to examine were those of the stomach. Now the stomach, I looked at the stomach many, many times. Here is a drawing of what I found. This is the stomach. Remember, I was somewhat of an artist, so here is the intestine. Okay? This is going toward the mouth. This is going toward the anus. Here is the stomach. Most of the cells of the stomach were like flat paving stones. But amongst these were these spherical objects. I've labeled them Proteosoma coccidia because I didn't know what they were. They contained actually malaria pigment, but they were clearly the parasite. So I had found now the development of this malaria parasite in the mosquito, beyond exflagellation, and on the stomach wall of this mosquito. I knew now that I had succeeded in filling in a gap in the transmission of malaria. And that afternoon was very hot, very overcast. And I remember opening the diaphragm on the microscope to get enough light in and see it as clearly as I could. And the drawing shows what I had seen. I recognized this was the malaria parasite because it had this pigment. And I decided to write my wife a little poem. This day relenting God had placed within my hand a wondrous thing, and God be praised at his command. Seeking his secret deeds with tears and toiling breath, I find thy cunning seeds, O million murdering death. I know this little thing a myriad men will save, O death. Where is thy sting, O victory, O grave? So here was the clue. The mosquito, after feeding on infected blood, the crescents, about 4 or 5 days, had developed these bodies on the wall of the stomach, and we call these the oocysts. I reported my findings to Manson, and they were published in a little journal in the British Medical Journal, and I entitled it, "On Some Peculiar Pigmented Cells Found in 2 Mosquitoes Fed on Malaria Blood," and it appeared December 18th, 1897. Now, I knew that I could wrap up this work in a matter of weeks, but then I was dealt a very unfortunate blow. The Indian Medical Service ordered me to proceed immediately to Calcutta, where there was an outbreak, I think it was of cholera. And in Calcutta, there really were very few patients that had malaria. I flooded an area and started to grow mosquitoes and so on, but there was really nothing to feed on. So even though I was given a laboratory, which consisted of a couple of rooms, an office, a cool, shady veranda, and I hired 2 assistants. One of the assistants, well, I actually fired the older assistants because these old people, they're really not much use. So I hired 2 younger men. One was named Purbana, and the other one was called Mohammed Bucks. And Mohammed Bucks was a rascally fellow, and I said to myself, anybody that's that rascally must be intelligent. Since there weren't a large number of malaria cases in humans, I turned my interest to malaria in birds. It turns out that malaria does infect birds. And so I set out a couple of cages covered with mosquito netting on some old beds outside, and I examined these birds. After a while, after uncovering them so the mosquitoes could bite, I examined the blood of these mosquitoes, and I found that some of them, in fact, had parasites, malaria parasites, inside red blood cells. And then what I did was I took these infected birds, sometimes they were larks, and sometimes they were sparrows, and I used clean mosquitoes, mosquitoes that I had reared in the laboratory, and allowed them to feed on the blood of these infected birds, and I found these same oocysts after about 4 or 5 days. Now the question was, what happens with the oocyst subsequently? How does one become infected from the stomach? And so after about 7 days, I made another dissection of mosquitoes that had fed on these crescents, and the next slide shows. I was examining the head region of these mosquitoes, and I saw what appeared to be a gland in the head, and I said to myself, this must be the salivary gland. Remember, these mosquitoes feed, and they have to prevent the blood from clotting. And they use something in their salivary glands to act as an anticoagulant. But here, in the mosquito salivary gland, were these rods. And these were obviously the infective stages in the mouth parts, in the salivary glands of this mosquito. So what happens is that the oocyst bursts, which contains some of these rods, and then these rods move into the salivary glands of the mosquito, where they sit until that female Anopheles mosquito bites once again. So on July 14th, 1898, I finally had something of value. I found the infective stages in the salivary glands of the mosquito. And these infective stages we call sporozoites. Pardon?
What was the infective?
The infected stages are called sporozoites. So you have an oocyst in which the sporozoites develop, and then the oocyst bursts. liberating the sporozoites, and the sporozoites find their way to the salivary gland, where they sit until the mosquito bites. So a mosquito, in order to become a transmitter of malaria, has to bite twice, once to become infected and once to infect. Due. So it was inoculation, not ingestion. And on July 25th, I sent off this telegram to Sir Patrick Manson, telling him of my success, that I had worked on this project now for years and years. And then on August 13th, Mohammed Bucks and I released all our poor little feathered prisoners. May their little souls be up in heaven, where there's flowers for them and berries in some avian paradise. Now, I want to show you one other picture, because you've heard me talk about Mohammed Bucks. I want to show you what Mohammed Bucks looks like. There he is, this rascally fellow, Mohammed Bucks. Here I am, pith helmet in hand, and here's my darling Rosa, my wife. And here are some of the cages of these that we held the infected birds in. And you see some of them are covered by mosquito netting, and we would put fresh mosquitoes underneath these nets so that we wouldn't have any contamination from the outside. And then we would have a clean experiment. Nice looking woman, Rosa, right? Okay.
What did Mohammed Bucks do again?
Mohammed Bucks was the rascally guy that we just worked for me. A smart student. But we have to give him credit. We have to give everybody credit in this world. But let me digress now. I have to speak about some Italians. The Italians are Battista Grassi, Amico Bignami, and Giovanni Bastianelli. Some have referred to my disagreement with these Italians as the Roman quarrel. Now, the word quarrel does not begin to describe these cheats, these liars who tried to steal my work, the glory of the discovery of mosquito transmission of malaria. They wanted to steal it for themselves. Now, as I've said, malaria was known in ancient Greece, and it was also particularly abundant in and around Rome. And the reason, of course, that we call this disease malaria is because it comes from the Italian word malaria, bad air. Now, what Grassi and his company did was they read my work on malaria described in birds, and they identified that there were two tasks ahead. What was the mosquito that transmitted the disease in humans, and what was the progress of this disease in the mosquito vector? And they had plenty of mosquitoes in around Rome. They called these Anopheles mosquitoes zanzaroni in Italian, and they, of course, had all of the characteristics that I had described earlier. And Battista Grassi recognized full well that Culex mosquitoes did not play a role in human transmission. That only in areas where Anopheles was there human malaria. Now, what did they do? In short order, they had described the development of the malaria parasite in the Anopheles mosquito, and they also described the development of the parasite in the human. They duplicated my work exactly, but they never gave me any credit. They scarcely mentioned my discovery, though they had followed my procedures step by step. It was piracy, not plagiarism, and that bastard, Grassi, He never even apologized and gave me a notice. His assistants, of course, did. So although I did not demonstrate the transmission of malaria in humans, I had set the stage for understanding that transmission. It was I who flagged the dapple-winged mosquito as the transmitter. And Grassi's contribution was simply to recognize these and call them by their name, Anopheles. Grassi and I were not co-discoverers, as he would like you to believe. I was the explorer at the helm of the ship, and the Italians simply rode on the decks and helped themselves to make a landing. And so it's fitting, I believe, that I received the Nobel Prize alone, and Grassi and his associates got nothing. Now let me show you what the life cycle of malaria looks like at the time of this discovery. So we have these parasites growing in the blood, destroying these red blood cells, producing anemia and death. And here they're not shown as crescents, but you have to imagine these as crescents. These crescents are found also in the blood. And then what happens is a mosquito, a lady mosquito, the femme fatale, bites and takes up all of these stages, but the only ones that survive in her stomach are these crescents, male and female. And the male shoots out these whips, which we call exflagellation, and they fertilize the female gamete, and then it becomes worm-like, and it passes from the inside of the stomach through the stomach wall and insists on the outside of the stomach wall. And what do we call that stage? Oocyst. Oocyst. Oh, right. Okay, so it's an oocyst. And you see within it, it begins to divide up into stages that are these rods that I've shown you. What do we call those? Those are called sporozoites. And these sporozoites eventually rupture the oocyst, and they make their way to the salivary glands, and then the lady mosquito, with her sporozoites in her salivary glands, feeds again and causes the disease. Now, there is one further complication, and that is, how do these parasites get into the blood? These sporozoites cannot directly penetrate these red blood cells. You see there's a gap here, and it's shown intentionally. And it took until the 1940s to discover where these sporozoites go before the parasites appear in the blood. And the next slide shows the complete cycle. The sporozoites make a beeline for the liver. They get into these liver cells and they multiply. So there is a development outside of the red blood cell first. And then for some reason that we still don't understand today, these infective stages that burst out of the liver begin to populate the red blood cells where they set up this division. And they never go back to the liver. So it's a one-way traffic pattern. Sporozoites injected, get into the liver, multiply, get into the red blood cells, and go back into the mosquito. So there are 3 cycles in malaria. 1 in the liver, 1 in the blood, and 1 in the mosquito. Almost at the end. Now, my discovery of the mosquito as the transmitter of malaria opened up new prospects for control. Control of this disease and other diseases that were carried by mosquitoes. In fact, the rest of my professional life was devoted to sanitation, getting rid of the breeding grounds of these mosquitoes so they could no longer act as vectors. Sometimes these destructions of the breeding sites were effective, and other times they were not. One of the great successes of the break in the transmission of malaria was in the construction of the Panama Canal. Were it not for sanitation and eradication of mosquitoes during the construction of the Panama Canal, it could never have been built. So I'm rather proud of my contribution. So now you ask the question, why is there so much malaria today if we have so much knowledge about this disease? Well, first of all, the parasites have a kind of plasticity. When you subject them to drugs, they are able to mutate, and they are able to resist the onslaught of these drugs. So we have to have new drugs each year, or each 5 years, or each 10 years, drugs that will be effective against the parasite. Second, we have another problem, mosquitoes. Sometimes you can drain swamps, sometimes you can spray insecticides. Insecticides are usually quite effective. But as you probably know, just like the parasite can become resistant to the drugs for malaria, so the mosquito can become resistant to DDT and other insecticides. So we have insecticide resistance. And the third is that in most of the countries where malaria is present, there are economic constraints. These are poor tropical agricultural countries by and large. Although, if you look at where malaria was present before the eradication campaign, this is just to show you that in 1923, here in the United States, in Mississippi, isn't that where Trent Lott came from? Poor Trent Lott. Okay, you see that there's a mosquito calendar, so that people would be forewarned. Stay away from mosquitoes between April and September, because those are the mosquito months. Next slide. Now, much of this tropical area still has malaria. And of course, they have lots of people. And so you have transmission continuing in these areas, and that's why we continue to have malaria in the tropics by and large. You know, Shakespeare, probably the greatest writer in the English language, knew that to have malaria was a fatal curse, and he gave voice to this in Caliban's Curse. You know, I too am a writer, although not so well regarded as Shakespeare. But is it merely a coincidence that both Shakespeare and I recognized the importance of malaria? And now I will say cheerio.